Guadayol, el doctor Òscar Guadayol, és un vell conegut de la casa. Va començar aquí, que el vam una mica enredar per fer pràctiques això de la carrera, per fer cosetes. Després es va deixar enredar per fer el servei social substitutori aquí, quan encara era obligat a fer la mili. Doncs ell va fer el servei social substitutori aquí i el vaig tornar a enredar una mica. I després es va deixar tornar a enredar perquè fes la tesi, quan jo, conjuntament amb la Cèlia Marrasser, vam ser els directors de la tesi de l'Òscar, quan jo no era ni tan sols Ramon i Cajal encara, o sigui que el nivell de confiança que ens va donar, almenys a mi, va ser molt gran. I després el seu periple ha tingut postdocs i contractes postdoctorals i altres als Estats Units, a Oregon, a Hawaii i després a Anglaterra, primer em sembla que va ser a la Universitat de Hull, no? Sí? Sí. I després a la Universitat de Lincoln, on encara està, i properament estarà a l'IMEDEA treballant amb l'Idan Tubal. El vam enredar a fer cosetes de turbulència, quan això era un tema complicat, mesures de turbulència al laboratori, experiments amb turbulència, també anàlisis de moviment de bitxos petits i tot això, i veig que al final, després de molts anys, algunes d'aquestes coses estan transposant amb els seus interessos actuals de mirar què li passa a les cèl·lules, i quan parlo de cèl·lules em dic bacteris i fitoplàncton petit, i la seva interacció amb el medi a nivell celular, d'acord? Amb el medi físic, amb el medi químic. I suposo que, almenys en part, la seva xerrada anirà d'això. I bé, sense més preàmbuls, doncs us deixo amb l'Òscar, que el títol promet moltíssim. Moltes gràcies.
Només una petita reflexió per començar, que jo no sé qui va enredar aquí, però bueno. Thank you very much for this wonderful opportunity to come back to... No em podré moure? No, okay. So thank you very much for allowing me to come home. Before I start anything, I would like to allocate the blame where it belongs. So if for whatever reason you see something that you think it's cool or you like or it's, it's a nice idea, it's all mine. Anything wrong, you can blame these guys, the people, past and present members of the Physical Ecology Lab in particularly, and then our collaborators in Scripps, in the University of Nottingham and in the University of Glasgow. Now that's out of the chest. Today I'm going to talk to you about microorganisms, about bacteria, about phytoplankton, and about how they live and how they experience their environment, their physical environment, and how it can affect the way their strategies for living and the way they move around and they acquire their resources. So it's a story that mostly I will explain it exactly or almost exactly the opposite way as I actually experience it, no? Because usually you end up with the conclusions of your research much later on, no? So you start with the really complicated things, and then as you think about them, they start simplifying, no? So there is one pearl of wisdom that didn't come to me very early in my career, which is that the physics that microorganisms in the plankton experience are quite different from what we are experiencing in our everyday life. So we don't have developed an intuition for what it is to be a planktonic microorganism, no? And this is, I think, the most striking example I could find. This is from a paper by Purcell in 1977, where he calculated how long would keep moving a bacterium after it stopped, moving its flagella. And it turned out that it would be about 0.1 Armstrongs in distance. That is 1/5 of the diameter of a hydrogen atom. This is completely different from what we experience in our everyday life, where if we start running and we decide to stop, we will probably crash. We will keep going and we will lose control of our trajectory. So it is very different to be a microbe. And this is all encapsulated in the infamous Reynolds number, which is basically a ratio between the inertial forces and the viscous forces. Okay? For very small organisms, the Reynolds number is very low. So the viscosity is way more important than inertia, no? So the key is, this is another way of looking at it. This is where we are here. There are, among living organisms, there are about 14 orders of magnitude in Reynolds number. Bacteria, phytoplankton are within what we could call the creeping flow realm, where there's no inertia at all whatsoever. How can we better understand why is it so different to be a microorganism? There's several different processes and mechanisms going on, but I would focus on these three very important and interrelated concepts, which are viscosity, Brownian motion, and molecular diffusion. All of them are concepts that are dependent one on the other, and they dominate microbial life in everywhere, but most particularly in the planktonic world. So viscosity, just to be a little bit more precise, what it is exactly viscosity? Viscosity is a measure of the resistance that a fluid offers to stress, to a force, okay? So if we have two plates and in between the plates we have a fluid and we move one plate and the other we keep it fixed, we develop a gradient of velocities, and the resistance that we encounter as we move the plate is directly related to what we call viscosity. It's basically something that arises from the molecular interactions. Okay, so as we get smaller and smaller, molecular interactions become more important than inertia. Why is it important for microorganisms? There's many reasons, but one could be, for example, that what we saw before, that bacteria cannot coast, they stop immediately, no? This is more anecdotal, no? But maybe more important for our interest is that the higher the viscosity, the higher the resistance to motion. So it means that if they want to swim, they have to invest more energy, for example. They will not be able to swim as fast. What else? Brownian motion. Brownian motion is this kind of wiggling movement that we can see in very small particles under the microscope, no? And it's basically caused by all the molecules in the water or in the fluid bumping around the particle, no? So what we see microscopically is just a random walk of these particles, no? This is so important that we have to use an equation made up by Stokes and Einstein, where we can relate the diffusivity of a particle to so how fast and how far they move, to the temperature, the size of the particle, and the viscosity of the fluid. So as you can, again, you can see the Brownian motion is related to viscosity. What are the implications for microorganisms? Well, I think that the two most clear ones to me are one, that a small microorganism in the plankton cannot stay still. It will keep moving. And so, for example, we can calculate that a bacterium the size of an Escherichia coli would move in 1 second the equivalent of one radius. Okay, so they keep moving around even without any active motion. The second thing, even more interesting to me, is the fact that they cannot swim straight. They cannot decide, okay, I'm going to go there where there is this nice fat phytoplankton cell and start eating. Because the moment they start walking and they lose direction. So this has been, we can calculate this, for example, for the same bacterium, moving at about 10 body seconds per second, it would lose directionality in approximately 10 seconds. The smaller you are, the faster you lose your sense of direction. No? So this is an extremely hard challenge for such small organisms, because how can they actually find a nutrient source if they cannot keep a straight path? And they have come with a quite clever way to solve this challenge. This is what we could call the biased random walk. So what they do is they run for a stretch, like usually about 1 second or 2 seconds, so a shorter time, so that they not lose significantly the direction, and then they stop. And then they can even let Brownian motion move them around so that they are directed to a random position, or they can start wiggling the flagella around so they actively change the position. No? And so the most common typical outcome is this run and tumble motility pattern. No? So they run, change direction, run, change direction, run, change direction, run. When they find, when there is a gradient that is interesting to them, notably a chemical gradient, so what they will do is that if they keep monitoring the concentrations of the nutrient, and if they see that the concentrations are increasing, they say, okay, maybe I should keep running a little bit longer here. If, on the contrary, concentrations decrease, they shorten the runs. It's that simple, but the outcome is that they can quite efficiently detect and track nutrient gradients in what is called chemotaxis. That's going to be important later on in the talk, because I'm very interested in this process. And finally, the third concept I would like to introduce is the molecular diffusion. It's very closely linked to Brownian motion, and in fact, I have here the same Stokes-Einstein equation. And at the macroscopic level, it can be described by the also very well-known Fick's first law. No? But basically, what it means is that molecules will move also by Brownian motion. And so they will diffuse, not, their movement is not linearly related to time, but to the square of time. So they take a very long time to diffuse very long distances, but they diffuse very short distances in extremely fast. And this is a concept that it's probably very well known to makers of perfumes or these kind of things, no? Because it is what drives our sense of smell, for example. Okay, how is this relevant to microorganisms in the ocean? Well, the most important thing here to understand is that all, let's say, all mass exchanges are dominated by molecular diffusion. So bacteria and microalgae that acquire dissolved molecules will be always limited by, ultimately, by molecular diffusion. It doesn't mean that turbulence or flow in the water cannot affect this by, steepening the gradients. But at the end of the day, any molecule delivered to the surface of an osmotrophic cell is going to be delivered by molecular diffusion. So, I think if you needed any convincing, I think I made my case on studying. If we look at the viscosity in the ocean at large scales, we see that it's relatively invariant. And what's even worse, from my point of view, it is perfectly constrained by equations relating it to temperature, salinity, and pressure. So we can just build this kind of global map of viscosity just by knowing temperature, salinity, and pressure. No problem. And that is the reason, I think, why in oceanography this question has been somewhat neglected. But we know that seawater is not pure. So these equations are applied to just pure seawater, not pure. But the more we know, the more we study the chemistry of seawater, the more we realize that it is an extremely complex system where we have a lot of different organic substances, like exopolymeric substances, gels, microgels, colloids, transparent exopolymer particles, mucus. It has many different names, no? And with a range of sizes, well, several orders of magnitude in range. All these molecules are coming from biological activity. A lot of organisms will release all these molecules, no? And most importantly to me, these may affect the physical properties of water. Just two examples. This is a very well-known fact that has been researched mostly by Ian Jenkinson in the 70s and 80s, but it didn't take much traction. No? There's here two studies I would like to show you. One is at the mesoscale study where they looked at the chlorophyll and the seawater viscosity and during about 4 days, right? And you can see there is a bloom in the chlorophyll here, which is more or less matched by an increase in seawater viscosity. So this is the background seawater viscosity. What we should be expecting using those equations I was telling you about before. This is what we find, that the viscosity can be easily, at these scales, 4 or 5 times higher. Another one, this is at fine scale. This is the scales here are from 0 to 45 centimeters. And here you can see the chlorophyll. I'm sorry, but the quality is, it's an old paper, I guess. But you can see that you have this patchiness, which is quite well known in chlorophyll, which is very well matched by the patchiness in viscosity at these scales. So it's obvious that viscosity changes way more than we could have anticipated. But what about the scale that is most relevant for microorganisms, the microscale, what they experience in their everyday life? Just to give you a little bit of background, in the last 20, 30 years, there has been kind of a change in paradigm about how we should be looking at this microscale, no? From starting with Azam's hotspot, where he envisioned that the the microscale was way more complex chemically and biologically than what we thought before. Adding then the Stocker's sea of gradient, where he envisioned that any microorganism would be kind of leaking away nutrients and creating these gradients of viscosity, of nutrients that could be used, that were used for bacteria to navigate their environment. So, so adding to this kind of emerging paradigm, we think we may also add the viscosity, because all these chemicals that are being released by microorganisms can also affect the physical properties of water, no? So we think our hypothesis is that there are microscale viscosity gradients that coexist with, overlap with, and even modify the chemical gradients. I have to say that this is not a very original idea, because the idea in some form or another has been suggested several times in the past few decades. No? So I put here a few quotes, but I'm just going to read the first one from Azam and Smith in 1991, which is the first one I could find about this. It says, "As one moves away from the surface of the phytoplankton cell and into the surrounding water, the polymeric matrix in the seawater would become less concentrated and less viscous. One might even hypothesize that bacteria use viscosity gradients to sense their environment and respond by making metabolic decisions." So it has been suggested several times that at the microscale there are viscosity gradients, but so far nobody had actually proved it until we came out with this really cool technique called microbiology. The technique that is vastly used in biophysics research and biomedicine, no? And it takes advantage very cleverly, I think, of one of the concepts I was mentioning before, the Brownian motion. No, Brownian motion of a particle is related to the temperature of the fluid, the size of the particle, and the viscosity. This is the Stokes-Einstein equation again, no? So if we can control for the temperature and we know precisely the size of any particle, we can calculate the viscosity in which this particle is moving. So we took this kind of concept a little bit further, and we decided to seed our samples of interest with lots and lots and lots of small fluorescent beads, of which we know exactly the shape and exactly the size. These are the same beads that you could use for flow cytometry, for example, or for grazing. So let me move. So each one of these red things that keep appearing is just we tracking one of these beads, no? So we can track the beads, characterize their Brownian motion, and from here we can extract the local viscosity of the bead. And using this, we can generate this kind of maps, which I will show you some of them now. So here we have the viscosity in colors, in different colors. And yeah, each one of these pixels in the map is about 1 to 2 microns in size. So we can do this now. We have this really cool technique. We decided to apply it to a bunch of different microalgae to see what was their environment around. No? So we had all these cultures of phytoplankton, of diatoms, dinoflagellates, and others. And then we seeded them with these beads, with these fluorescent beads. And then we look at the microscope microscope with fluorescence and epifluorescence and phase contrast. And so the results were published in this paper in last year. I'm just going to give you a brief overview of what were the main results, which were basically one that we found extreme heterogeneity in viscosity at these scales. We also found very high viscosities inside aggregates of marine particles. Then we also found that the phycosphere, which is this kind of the region surrounding phytoplankton where most of the interactions with bacteria and other microorganisms occur, is very viscous. And finally, we also found that when phytoplankton dies and lyses its contents, viscosity increases. Let me be a little bit more detailed. So we found, this is a random map of one of these cultures of viscosity. Here we have a cell, but otherwise there's nothing else here. When you look at the image, at the phase contrast under the microscope, you don't see anything here. But then the microbiology reveals this kind of very patchy structure, where viscosity can be easily 5 times higher than in water. And the size of these patches is a few micrometers, So obviously there's a lot of mucus or tape or whatever that is there altering the physical properties of water, and we were not aware of it. 2, we used this, how do we call this? This rolling device to generate marine snow. And then we seed it, we take the sample very carefully and we seed it with our beads. And we also found extremely high increases in viscosity, up to, I think it was, the record was 80 times the viscosity of seawater. Now, these are hotspots for microbial activity, and you can imagine that the fact that the viscosity is much higher here, it might change the whole dynamics of these kind of systems. And then the phycosphere is viscous, So we deem this the viscosphere. In many cases we found that around the cell there was an increase in viscosity, nonlinear, usually exponentially shaped, which could extend about a few micrometers, even 10 or 20 micrometers away from the cell. And finally, we also found we induced the lysis of phytoplankton cells using UV, and we found, so this is an example here, you'll see at 0, and then as you can see there's a blob originating that moves away. So we found this is the viscosity versus the distance from cell. In blue is before lysis, in red is after lysis. So by bursting, they release a lot of the cytoplasm, which is way more viscous than water, and they change again the properties of the whole frame, which is actually quite a lot. How common are these viscospheres? Well, we found in our experimental system that it was very common. This is a kind of a summary plot where we have the relative viscosity at a distance of 2 micrometers from the cell wall, and then we have the volume of the cell, and then we have here different species, and this represents the extent of the viscosphere, okay? From non-significant to 4 micrometers all the way up to more than 32 micrometers away from the cell. So all the ones that are darker are significant. So we found significant increases in viscosity in all these examples. Surprisingly, we didn't find any relationship between the relative viscosity and the volume. We would have expected this to have some relationship, but we don't find it. All this has been published and with all the collection of this kind of maps here if you are interested. So we've found all these cool facts that were suspected but mostly unknown. But the question to me now is, okay, but what is the ecological significance of all this? How important is this? No? And this is our struggle now. No? This is where what we are working on now. So, just not with Stuart Humphreys, we have been thinking a lot, trying to make up a framework of possible effects of the presence of this viscosity heterogeneity in the ocean. And the truth is that because viscosity affects so much of any microbiological interaction, the effects can be quite wide, no? Starting from physical effects, so for example, it can affect the turbulent scales, no? Because it may affect at which the size of or the length of the Kolmogorov scale, for example, or the Bachelor scale. Viruses, it can affect virus infections because the relative motion of the host and the virus is going to change, so it can be, maybe we think it could be a strategy by some phytoplankton to minimize the risk of being infected by viruses, for example. It can affect the microbial loop, and I will go a little bit, I will explain a little bit about this later on. But it could also affect the downward carbon flux because it can affect first the dynamics of the marine snow, how they remineralize and how they are colonized by bacteria, for example, but also can affect the speed at which these aggregates sink, for example. They can also affect symbiotic relations, for example, with corals and microalgae or bacteria. I'm going to focus for the rest of the talk about this, about the microbial loop, more specifically about the interactions between bacteria and phytoplankton, no? And centering in this EPS, the exopolymer substances that phytoplankton release, no? Over the years, there has been many different hypotheses as to why algae would be releasing these exopolymer substances. No, we know that they can affect the attachment of these cells with other surfaces. So they may affect the formation of biofilms or the formation of marine snow ultimately. It has also been suggested that it's kind of a metabolic excess waste products, that they don't serve any function, that they are just there because this is cheaper than just shutting down the whole machinery. Enhance it forward scattering of photons, so they can redirect the photons and increase the efficiency of the photosynthesis. Protection from grazing or from viruses. And also protection against physical insults. To me, the ones that we are thinking about is they could enhance microscale gradients, help absorbing nutrients, and also help conservate exoenzymes. So hindering the diffusion away of exoenzymes from bacteria. Okay? So, about this one, enhancement of microscale gradients. I mentioned before that the molecular diffusion was very related to viscosity, no? And so our first thought would be, okay, these gradients around cells, what they will do is slow down the diffusion of nutrients away and to a phytoplankton cell. No? And so we use these dun chambers to create a kind of a gradient of viscosity, and we look at how chemical, in this case fluorescein, which has a diffusivity similar to any nutrient in the ocean, diffuses away when there's only a chemical gradient, or when we overlap to this chemical gradient a viscosity gradient. And as you can see, the resulting profiles are dramatically different. Here is again looking at the time. This is time. This is the distance in this axis, no? And this is only chemical gradient, and viscous gradient. You can see with time, very quickly, the gradient, the chemical gradient is established when we have viscosity. When we don't have viscosity, it sticks to the source. It doesn't diffuse away. Trying to understand this process more in our system, we developed some COMSOL models. Here we have our spherical cow, here, the phytoplankton cell, at the initial concentrations, and here inside we assume that the cytoplasm is full of viscous content, no? And then we assume that it bursts. And then after 5 minutes we have two scenarios. One in which we assume viscosity is not changing, and this diffuses away quite fast. And two, when we assume that viscosity is changing, and then we see that the chemical gradient again doesn't move away. Recently, Bryce Inman in Script has published this paper where he hypothesized the fact that actually having this mucus layer around the cell can be detrimental to phytoplankton cells because the uptake rates decrease. So this is the viscosity profiles, and this is the diffusivity resulting from these viscosities, and this is the concentration of the nutrients resulting of these. And the point, the message here is that these viscosity gradients have to be really useful in some way if we can assume that the fact of having them can be in some ways hindering the growth of this algae. Again, this is hypothetical, and I'm not quite sure that this is going on, but this was published this year in LNO Letters. Then, so we know about how nutrients are going to be affected. What about bacteria that want to track these nutrients and go to the phytoplankton cell? So to understand this, we took more of an experimental approach. We exposed a population of Escherichia coli to solutions of different viscosity. And then we track them. We take videos of them moving, and we track each one of these, and we looked at how they were swimming. No? Here, incidentally, here you can see the kind of run and tumble typical pattern of bacteria, of palmitotic bacteria. Okay? And this is the outcome. This is a histogram of, this is microns per second. This is an histogram of swimming speed of E. coli. It's about 20 microns per second, which is exactly what we should expect. And this is a kind of a polar histogram of the directions of the tumbles, of reorientation, you know? And you can see it's more or less uniform. So when they tumble quite randomly around. No? What happens when we increase viscosity is that the speed dramatically decreases because the resistance, the drag that they are experiencing is way much higher. No? So this, in this case, is 1 order of magnitude lower. And then also their directionality is completely changed. So instead of the tumbling, instead of being completely random, they cannot turn completely, because again the viscous drag doesn't allow them to completely turn. So their pattern is changing. So instead of doing a random tumble, they do a more directed tumble. Once they tumble, they keep going towards the same direction they were going before. And this is kind of a summary plot where you see increasing viscosity, swimming speed decreasing, and this is a metric of this tumble constraint that I was telling you. And again, as viscosity increases, they are more and more constrained in their directionality. So we took all this data and we built an individual-based model where we simulated hundreds, thousands of bacteria swimming and doing chemotaxis, and we overlapped to the simulations different gradients of viscosity. So here, for example, we have a gradient of viscosity, but also of nutrients and nutrients. No? Even in this very short clip, you can see how they are very fast here, very slow here, and the end result of this is that there's no chemotaxis involved here. But ultimately, this leads to bacteria without any behavioral change accumulating near the viscous source. So we did all these, ran a series of simulations using this model. This is the results from in a geometry where we have a phytoplankton cell of about 25 micrometer releasing aspartate, which is a strong chemoattractant for E. coli. And then, sorry, the colors are a little bit wrong. The blue line here represents the viscosity profile. The yellow one here represents the aspartate profile when there is a viscosity profile. And the red one is the gradient, the nutrient gradient when there is no viscosity. Okay, and this is the result. This is the bacterial abundance normalized by the background abundance. So it should all come down to 1. And what you see is that when there is no gradient, it's always 1. When there is a chemical gradient only, there is this nice peak here, and then it decreases. But after this is after 5 minutes, the concentration in first 100 micrometers around the cell can be easily 10 times what it is in the background water. When you have a viscous gradient on top of the chemical gradient, you see a massive increase in viscosity, and what's more important, they can get closer and closer to the cell. No? And what I was telling you about, when there is no chemical gradient at all, just viscosity, even with only viscosity, you also see an increase in an accumulation of bacteria. It's a physical mechanism, but it's very interesting, no? So it means that they don't really need to spend their money in excreting all these fancy molecules that will attract bacteria. They can just rely on the viscosity to make the job, no? Okay, so to wrap it up a little bit, these are the take-home messages, no? How am I doing? Viscosity is heterogeneous at the microscale. We can see that nutrient hotspots coexist with viscosity gradients, which is something that was suspected, but we didn't know before that. At the most basic level, viscosity affects the diffusion of nutrients and the plankton motility, no? But this is at the most basic level, and then we can think on ways in which all this scales up to have profound implications on how the planktonic ecosystem works. what about the future? What are we going to work on now? So one thing we are not discussing is we want to have a more detailed physical picture of these compounds. So we are going to look at the viscoelastic properties of EPS and also any anisotropy that we could find. We are also very interested in understanding what is the variability in the field of these. What is the ecological relevance? How do we find this kind of patchiness in the field? I don't know yet, but one of the things that we hope to be doing with Cesc and Montse is to take samples from out there and measure and produce this kind of viscosity maps. Then we want to do experiments with microfluidics, and we have quite advanced these, where we have designed these kind of microfluidics chips that can create viscosity gradients and chemical gradients, and so we can put bacteria in here and see what is the effect, no? See if whatever we found in the IBM model is actually real, no? And then finally, what I'm also very interested in understanding what is the size-dependent effect of viscosity, no? Because I've been picturing you quite simplified thing, no, here, which is that viscosity is just a linear thing, and it doesn't just one simple parameter. And the fact is, it's not. It changes a lot with the structure of the mesh of the polymers that are being released. It changes with the solvent. It changes with the temperature. But most important to me, it changes with the size of the probe. So, which means that what we were measuring with all these maps was the viscosity that these beads that were 0.25 micrometers in diameter were experiencing. But it doesn't mean that the molecules, the nutrients, are experiencing this effective viscosity. No? So here we have the size of the particle against the viscosity that is actually being experienced by these particles, no? And it's quite changing, no? So that got me thinking, and I am going back to the beginning, to the title of this talk. What if this EPS is actually an engineering tool, something that phytoplankton release to modify, to control their environment, no? Because if you think about it, it can change the physical structure, we know that, but it also optimizes the recruitment of chemotactic bacteria, right? Because of the viscous trapping effect, and because it steepens the nutrient gradients also. It also reduces the diffusion of large organic molecules that bacteria would be interested in. So maybe it's creating like a nice environment for bacteria to live there, and so that promotes the healthy relationship between bacteria and phytoplankton in the phycosphere. But if as we were saying before, if it doesn't affect too much the diffusivity of inorganic nutrients, maybe it doesn't have any impact on the uptake rates. So what I'm proposing here is that this is kind of a size of resource partition of resources, no? Size partition of resources, or size fractionation, if you want. So with just one simple tool, you can solve different problems. I wanted to finish acknowledging Mikael. I just wanted to say that he was, I think, the first person that instilled in me this notion that planktonic organisms, yes, they are at the mercy of the physics, of the large scale physics, but they are also extremely complex in their behavior, and that they can, they are not as simple as we thought before. And I think he was the first one to make me understand this, which is underlying the whole talk, if you realize. And that's it. Thank you very much.
Well, thank you very much, Òscar, for a great talk and lots of food for thought. So we open the floor for questions. Anyone?
Hello, thank you very much, Òscar. This was very stimulating talk. I must say that I passed the link of this talk to Ian Jenkinson, Lawrence Seon, and a group of people that are interested in rheology and the work you are doing. So if they are here, hello. Okay, then concerning the, I have plenty of questions that come to me, but then one question. When you mentioned size, you were thinking the size of the depth of the molecules of the cells. Why I put this question? It's because the interactions between the inorganic components, such as specifically calcium, and all those ions that are inorganic, with the interactions with organic compounds, also contribute to the increased bindings and maybe, let's say, not biologically stimulated formation of TEP. So, my question is if you are considering also those chemical interactions between molecules apart from the, let's say, the activity of the cells. Because when we think about lysis, lysis is a biological process. Mm-hmm. And then the components are released. But once they are released, there must be interactions in the chemical world, sensu stricto, that will also contribute to this viscosity. Mm. So it's like an overlapped mechanism that can be influencing on all the processes that you are seeing here. So the inorganic, organic bindings that contribute to the formation of TEPs, etc., in a maybe not living way. So is this the question? Are you also considering, or or maybe it doesn't play any role on those chemical interactions?
No, great idea, because I hadn't thought about this. It's as you know, it's a little bit farther from my field of expertise, so it's something that didn't occur to me. But yeah, I think it could be quite relevant. It would be nice to investigate this. So when we are talking about the size dependence,
yeah.
Let me go back. Here, this is viscosity,
yes.
And if you notice, these are logarithmic axes.
Okay.
And this is the size or the molecular weight of particles. So it means it's the diffusion of either a molecule or a particle. It doesn't matter,
okay.
No? So, in the same environment, in the same solvent, in which you have a matrix of wherever macromolecules you want, you could find that very small molecules go in between without, they don't feel any difference with water. So if you release here any calcium or whatever, it can go all the way to the other side very quickly.
Exactly.
So it doesn't slow down chemical reactions, I would expect. Whereas if you are a bacterium up here, or if you are a large molecule, then you will find a hard way to go all the way. So
Exactly. So what I mean is that this process will be interdynamic. The the size of the compounds, of the chemical compounds, will be nothing, it will be not only static, like a molecule has a size, but then those overlapping mechanisms of interactions with inorganics will make these molecules become bigger and bigger and bigger, so it will be one of the mechanisms in this very nice picture you are having here. This is the idea, that it will be a dynamic component also with the participation of these interactions between inorganic and organic compounds. This is one of the ideas that is stimulating this image.
I'll think about it, because I'm not sure I can address these kind of questions, but we can talk about it.
Thank you.
Anyone else?
Hello, Òscar. Many thanks for this inspiring talk. I have just a curiosity about the importance of temperature in this viscosity, and how should be the optimal temperature that will make that viscosity will be reduced.
Yeah. So, viscosity is there. It doesn't. The higher the temperature, the lower the viscosity. That's it. But what's important here is that the relationship between temperature and viscosity is different for different molecules. Okay. So if you have a gradient of viscosity because you have EPS or whatever, and you change the temperature, you can magnify or reduce the viscosity gradient, which has implication, for example, for nutrient uptake. Let's say, for example, if you increase if you increase the temperature, you may erode these viscosity gradients faster, and also erode at the same time the nutrient gradients. So the whole chemical complexity and physical complexity may change because we are changing temperature. But the relationship is very simple. It's just that it changes, the slope changes with different molecules.
Thanks.
Hi, Òscar. Thank you very much. This was an amazing talk, and we have discussed this a lot in the past, but now something new came to my mind when listening to you. Have you ever explored whether the vicosphere changes somehow the swimming pattern of, or the motion or whatever, of the organism producing it?
Yeah.
Because this would have also an impact in grazing rates, for instance.
Yeah.
If you are if the one producing the vicosphere is the prey, and then counter rates and all this one.
No, but we haven't thought about this and theorized about this. No, it's not just on the motion of motile particles, but also sedimentation. No, because effectively you are changing the area or the volume of the particle, so you are changing the physical properties. You are changing the drag that it's experiencing. No. So it must have an effect. It must have an effect. I'm not, I don't know, I would be a little bit surprised, because this is something that they can probably turn on or off, no? So there were lots of phytoplankton cells that would not have a viscosphere. So I would assume that they can, increase or decrease the production and the release of these substances depending on the circumstances. And in fact, when we were looking at just the heterogeneity of viscosity in the cultures, without looking at specific cells, we did notice, for example, that the heterogeneity was way higher in older cultures where it depends a lot on the physiological status of the organism. But yeah, we thought about that. It would be cool if we can somehow replicate this in an experiment, no? See if we can actually look at look at the motility with and without but the physics are quite clear about it. It should have an effect.
Hi, Oscar, very nice talk. I was wondering if you, because my understanding is that E. coli has a different movement strategy than other marine bacteria, and that it's not the common strategy in the ocean. Do you know why?
Yes, well, it's in fact I'm working quite a lot on that now. So the most typical strategies in the ocean are more like run and reverse, or run, flick, reverse, or so there's always a component of reversing, no? When you look at the so this is run and tumble, basically, no? But when you look at run and reverse, when instead of seeing this bump, what you can see is that it gets closer to the cell, no? And it has been hypothesized before that run and reverse, it's a good strategy to keep close to the nutrient source. What happens here? Imagine that you have a spherical cell here, and then bacteria swim in their pattern wherever it is, and if they swim fast, which marine bacteria tend to do, they can easily overshoot, and that's why you see that peak here, no? But having a run and reverse allows them to immediately backtrack themselves and go back to the nutrient source. So if you imagine this is the surface of the cell, they could be doing like this and keep right in the nutrient gradient.
And another comment, I was hearing you talk, I was thinking maybe viscosity has a role in the colonization of particles by bacteria that are not motile. Because we found in oligotrophic regions in the deep ocean that many particles have bacteria that don't have cells for motility. So maybe if they get viscous and in the deep ocean with the temperature they have higher viscosity. Maybe that's a way.
Yeah, because as I was mentioning, Brownian motion means that even non-motile bacteria will move quite substantially over time. So this effect here, it will happen. So this exact same shape will be found in non-motile bacteria. Mm-hmm. The exact same, because it does not depend on the speed of motion. The only thing that depends on the speed of motion is how long it will take to reach this kind of steady state. But yes, even non-motile bacteria will at the end accumulate around viscous patches. Yeah.
Okay. Thanks. Very cool.
Hi, Oscar. I have so many questions and comments. I don't even know how to start. The one that I never thought about is about this gradient trapping of the movement of bacteria. And you sort of said, well, you don't even need sensing, right? Of course there is sensing, because otherwise they would be trapped always by these chemicals. Either when they are good or when they don't want them, like an antibiotic or something else, right? They would never be able to escape that. And we know that bacteria do move away from chemicals that they don't like. Yeah. So, but of course it will be much harder to move away from a gradient of a chemical that they don't like than to be attracted to a chemical that they like or that's neutral to them. So it's harder to move away from if the gradient operates and always operates as a trapping mechanism. And this is something really interesting to explore, I think.
Yeah. Yeah, and it could be easily done. So we could easily do this exact same IBM, but instead of having a chemoattractant, you have a chemorepellent, and then you would see immediately if there is a
But my guess is the gradient still operates physically, so they have to do something extra to move away from it.
No,
I don't know if it has energetic costs or not, whatever.
I'm going to do it.
Yeah, so interesting. Then another thing is, global implications now with global warming. Viscosity in the upper layer should decrease, and this has lots of implications because, for example, the same energy that you put into to generate turbulence, now with a decreased viscosity, this turbulence reaches a lower scales, because it has implications in the Batchelor and the Kolmogorov scales and so forth. So this is really interesting because even if we have a calmer upper ocean, it will also be punctuated by larger storms and more frequent storms, and the effect of these storms will be able to go to lower scales and produce inertial motions there, or inertial effects, or inertial derived effects, or whatever. So I don't know if you want to comment on this, and even if changes are really small, because we're talking about small changes here in the viscosity and so forth, but we know that nowadays small changes at the global scale can have huge implications on biogeochemical cycles. So I don't know if you want to care about to comment on this.
Well, yes, we did mention all this in the paper, and it found some resistance because, yeah, it's hard to imagine how these kind of scales can be affecting anything else, right? I think the physics know perfectly well how these will affect at large scales, how changes in temperature will affect. I think they must have considered this because it's straightforward. But what we are adding here is what I was saying before, no? that the functional relationships between viscosity and temperature change with the chemical composition of the ocean. So if you have a diatom bloom, a phytoplankton or whatever that is increasing locally the viscosity, it will change the physics at that scale and higher up, no? And yes, I would like for physics to start considering this kind of biological variability as something that will ultimately affect the physics, no?
Yeah, but even without considering this, which will be probably the most important part, but just from the physical point of view, without any chemicals or anything in ideal water, if the viscosity gets a little bit lower, Kolmogorov and Batchelor scales go down.
Yeah.
And this can have huge implications.
And not just physics, but biology too, because...
And also related to that, then there's the deep ocean where you have pressure as well, and the pressure decreases viscosity. So things there are even more like...
And the pressure increases viscosity.
Increases viscosity, I'm sorry. Yeah, that's right. So the things are still moving slower there.
Yeah.
This might have also huge implications at the biogeochemical cycles.
In fact, there is a few, I'm not following this much, but I know there is a few papers that discuss the fact how increasing pressure affects a lot of different chemical reactions and biological interactions, and I am wondering if it's not because of this. That increases viscosity, so it changes the speed of any reaction.
And then lastly, something that's also really interesting in this global change that we're in, is that the increase of temperature, let's see, in air, okay, this will, what will it do? It will increase decrease the viscosity as well, right? Of the air.
The increase in the temperature in the
Let's see,
atmosphere.
in the water it will decrease viscosity because it increases temperature, and in the air it will
the same. Yeah.
What?
It will decrease viscosity of air.
Much more, right?
Yes, I think so. Yeah.
So in terms of fluxes of air to sea and sea to air, it might have, I don't know, I haven't done the numbers or thought about much, it just occurred to me when you were talking, that this might have implications there as well.
Yeah.
So I don't want to hog the conversation here, but
Yeah, yeah, of course you would know way more about this than me. But yeah, I guess that I mean, the relationship in the two different fluids with massively different densities is going to be different. So maybe the rate of change of viscosity is way faster in the atmosphere than in the ocean. Is that what you mean, right? So that may, yeah. It's, so maybe I don't know, to summarize a little bit, I think that I'm very enthusiastic about this because I realize this is something that, I'm not saying that there has not been work done before, no? But if you start thinking about what are the possible implications, it's hard not to find a new one every time you think about it, because it's pervasive. It affects the physics of, it's like temperature. Temperature is affecting everything. Viscosity is the same, right? It's something that affects, especially at these scales, everything. So, so I think we need to start thinking about viscosity also in the same way that we think about temperature as an environmental variable that will affect everything. With the added thing that temperature is not going to change at such small scales, whereas viscosity does. It just complicates things as usual.
as I said when you finish your talk, lots of food for thought. So does anyone have another question or comment? So thank you again.